So coming to gene mutations, now when it comes to mutations, mutations are nothing but they are sudden discontinuous variations. Okay. So when it comes to a mutation, mutation is a change. They're nothing but, when it comes to gene mutations, these mutations are sudden and they are discontinuous variations. Discontinuous variations in the genotype. So if there are changes in the genotype, of course, there will be changes in the phenotype as well. The genotype and phenotype, okay, respectively. So mutations are sudden and discontinuous variations. Variations are nothing but changes in the genotype and phenotype of an organism, okay, of an organism. And all this is due to changes in the chromosomes which have genes residing on it. So when it comes to mutations, as I said, there are certain changes in the sequence of the bases. All right. So because what is a gene? A gene is nothing but a sequence of bases. Correct? It's a sequence of bases which is responsible for a particular protein, and that protein is responsible for a phenotype. So example is, let's say I have gene 1, okay, residing on a chromosome or on a DNA. So what is a gene? It's nothing but simply a sequence of bases, right? So when it comes to this DNA molecule, let's say this is the gene for an eye color. And let's say sequence on the 5 prime 3 prime end is TAGCCTTAAC. Okay. Now let's say there is a mutation. So basically this particular sequence that you see from T to A. Now this is a gene which is responsible for a protein, and that protein is responsible for, let's say, black eye color or for deposition of black color in the iris. Now, whenever there is a mutation, we are learning different types of mutations. Okay. So mutation is nothing but changes in this entire reading frame or the sequences. So let's say instead of T, A, G, okay, and C, C, let's say there is one more G here which is placed. Okay. And then there is, let's say, C, and then T, T, A, and C. So can you see here the changes what has happened? So instead of C, what you have is in the DNA, you have a G here. Okay. So this is a mutation change. So this entire reading frame or the sequence, what you see in the 5 prime, 3 prime end, this has changed. Okay. So this is a mutation. Just one example I've given you. So coming to gene mutations. Okay. So coming to gene mutations is what we are learning. So these are alterations or changes in the DNA because of change in the nucleotide sequence. Nucleotide is nothing but, nucleotide has a base. So every nucleotide is made up of, these are the building blocks of the DNA. They're made up of phosphates, then you have a sugar, and you have a base, correct? And these are the bases that I'm speaking about, ATGC. Now, gene mutations are nothing but they're changes in the DNA. They're alterations in the DNA due to change in nucleotide sequence, due to change in nucleotide sequence. Now, under gene mutations, what we are learning is, okay, under gene mutations first, we are learning point mutations, okay. Now, what is this point mutation? Now, if you have just one single base pair of the DNA, which has changed, now here see this example what I've given you, just one base basically has been changed, correct, in the DNA. So instead of C, what you have is G, which is placed. This is called as a point mutation, okay. So what is a point mutation? Mutations which occur due to change in the single base pair. So change in single base pair of the DNA. That's called as a point mutation, single base pair change. Okay, so this example what I've given you here. Now, under point mutations, basically what we are learning is, or gene mutations. So if there is just single base pair change, we call it as point mutations. All right. Now, what is gross mutation? Gross mutation would be changes in more than one nucleotide. So gross mutation would be changes in more than one nucleotide. Okay, that's called as a gross. Now we are learning under gene mutations or point mutations. Okay, we are learning point mutations basically. Let's see point mutations, what they can do. So there is something called as a frame shift mutation. Frame shift mutation. Okay. Now for this, I'll have to, for frame shift mutation, I'll have to tell you a little about, the sequences and the bases first. All right. So before we go in for frame shift mutations, I'll tell you about the DNA first. I'll just place a DNA molecule here. Okay. So this is my double stranded DNA molecule. And the sequences of the DNA molecule here are as follows. Let's say to start with, okay, I have this 5' 3' end, and I'm writing the sequence of only 5' 3'. Let's say to start with, I have A, U, G, okay, not U, sorry. Let's say I have A, T, G. Then let's say I have the next sequence, which is C, A, T, okay. Then let's say I have T, T, T. So let's say these are the sequences. So this is my DNA molecule, okay. And let's say the last one what I have here is again TGA. So this DNA basically on transcription is going to give me an RNA sequence. Okay. And this RNA will have now the sequence is 5' 3' end. It will have the same sequence as that of 5' 3'. Okay. But instead of T, there will be U. So I will have A, U, G. Then I will have C, A, U. Then I'll have U, U, U. Okay. And then I will have UGA. So instead of T, the RNA has got uracil molecules. Okay, they don't have thymine molecules in them. So when this DNA is giving you an RNA on transcription, okay, so this process here is transcription. So on transcription, okay, your DNA is giving you this entire gene basically is read into a simpler form, which is your mRNA form, messenger RNA form. So this is the entire sequence that you get. So instead of T, there will be U. So just the same 5' 3' end is copied, and instead of T, you will have U. Now, when this particular mRNA is undergoing translation, okay, when this is undergoing translation, now if you look at each of these, bases. Now, if you look at these bases, AUG, okay, then you have CAU, and you have UUU, and you have UGA. You have U, G, and A. Now, each of these triplets of the bases is called as a codon. So, what is a codon? This is codon 1, 2, 3, and 4. So, codon is nothing but it's a triplet of bases. Each of these triplets that you see, okay, they will code each of these triplets that you see, they will code for an amino acid. So this AUG, this codes for methionine, which is an amino acid. Okay. CAU will code for histidine. UUU will code for phenylalanine. Phenylalanine. And this UGA does not code for anything. So this is a stop codon. This is a stop codon. It does not code for any amino acids. So here, these are your amino acids. Okay. Now, AUG, if you see here, as I told you, this AUG, they're read in triplets, right? This is read in triplets, triplets, and triplets. So AUG, if you see, this will code for methionine. I'll write it as M. CAU will code for for histidine. UUU will code for phenylalanine. And this UGA is a stop codon. That means nothing else will be present. So this is going to be my protein chain. So this is going to be my protein chain. And the sequence of my proteins will be M, H. Okay. It's going to be M, H, and P, which is methionine, histidine, and phenylalanine. Now, this gene is giving me this protein. And the sequence of my proteins is methionine, histidine, phenylalanine. Okay. Now, if there is a mutation, okay, so this is my reading frame. What is my reading frame? How is my entire messenger RNA read as AUG, CAU, UUU? Okay. Now, let's say that there is a mutation in this DNA. So let's say that there's a mutation in this DNA, and let's say that instead of CAT, what you have is here CAT. Let's say there's a mutation in one of them. Okay, so let's say instead of TTT, what I have is here TTT or UUU. Let's say the last one, the last T that you can see. Okay, let's say there's a mutation here. So instead of T, okay, so instead of T, let's say I end up getting C here. Okay, so let's say there's a mutation, star for mutation. And let's see, instead of TTT, I have TTC. If there is TTC here, this sequence will be UUC, isn't it? So here this sequence instead of UUU, this is again going to be UUC. And UUC, this codes for phenylalanine. Okay. Now this UUC also codes for phenylalanine. If you see here, UUC, this also codes for phenylalanine. Now, though there is a mutation, okay, but UUC and UUA both code for phenylalanine. So don't you think the sequence of the protein will remain the same? Even if there is a mutation here, but instead of this P, you will have a UUC protein, phenylalanine only, but the sequence of the protein remains the same. So such mutations are called as silent mutations. So such mutation will be, what I first told you here, is a silent mutation. So what are silent mutations? There is mutation in the DNA, but it is not shown in the phenotype or in the protein because the protein is the same, M, H, and P again, because UUC also codes for phenylalanine. Okay, so these are silent mutations. So if you look at the sequence here, I've shown you, it's the same sequence, but instead of PTT now, you have GTT. G is the mutation. So instead of T, you have a G here. Okay. Now, because of this, the entire mRNA sequence will be AUG, CAU. Instead of T, just place uracil. This will be GUU and then UGA. Now, GUU codes for valine. Okay, so AUG will code for methionine, CAU for histidine, GUU for valine now. So if you look at this protein, this says M, H, and V, and this was M, H, and P, correct? So this is a different protein as compared to the original DNA sequence, hence this is a mutation. You have a different protein, hence a different phenotype. Okay, so mutations in gene cause a different phenotype. If if the mutations do not change the protein sequence, then we call them as silent mutations. But if it does, because of a single base change, we call it as a point mutation. So keep in mind, when it comes to silent mutations, okay, there is no change in the phenotype. But silent mutation, what example I've given you here, there's only one base pair which has changed. Okay, so this can also be a type of point mutation. But we call it as silent is because there's no change in the phenotype as such. Okay. Now let's come back to the frame shift mutations. Now, under this, what we are learning is deletion, okay, and second will be additions. So I'm showing you the same DNA sequence again. And let's say the sequence now is, okay, I'll give the same sequence. Let's say there is A, TG, CAT, TTT, and let's say there is TGA. Okay, this is my 5' 3' sequence, my DNA. And the RNA that I've now got is the 5' 3' sequence is AUG. Okay, that is CAU, UUU, and that is UGA. Okay, now I'm showing you deletions first. Okay, first we go with deletion. Let's say out of this entire sequence that you had here, okay, let's say there is a deletion, and let's say that CAG has been deleted. If this was the sequence, this would have been, your entire original sequence would have been this, correct? Your mRNA would have been would have had this particular sequence. Now let's say that because of a mutation, because of a deletion, let's say one of the base has been deleted. And what is that base which is deleted? Let's say this C has been deleted. Okay. So this C is now deleted. This codon is deleted. This particular base is deleted. Obviously, if this is deleted, the C will also not be present here, correct? So the C is missing here. Now, when it comes to the entire sequence on the mRNA, it's going to be read as, it is always read in triplets. So you have certain enzymes which are going to be recognizing, okay, they will recognize AUG as the first codon. Then they will recognize, because C is missing, they will go to AUU. Okay, they will read the next three. So this will be read as the second codon. Then you will have UUU, which is going to be read as the third codon. Then it will go on to the next. Okay, so let's say there is C present here next, C. So it will go to GAC, which is going to be read as the third codon, fourth codon here. Okay. Now, so AUG basically, let's say this codes for methionine. AUU now you'll have to go in for, correct? So don't you think the entire reading frame has got changed? So let's say AUU, let's say it codes for tyrosine. UUU, this codes for phenylalanine. And let's say GAC codes for one more amino acid, let's say cysteine. Okay, just an example I'm giving you. So don't you think this entire protein which is formed now, okay, this protein which is formed now is different? Because initially you would have got M, H, and P, but here it is M, T, P, and now C. So this is a different protein. Now, because of a deletion, can you see here? Because of a deletion, don't you think the entire reading frame has got changed? Instead of AUG, and this was next was CAU. Instead of CAU, it is now read as AUUU. Are you getting it? It is read as AUU. So the entire reading frame has got changed. The reading frame is changed. Such because of, okay, the reading frame has been changed. Such a mutation is called as frame shift mutation. Okay, so the entire reading frame is changed, because of which we call it as frame shift mutations. So deletion results in, I can say that because of a deletion of a base pair, because of a deletion of a base, this leads to what kind of mutation? Frame shift mutation, wherein the entire reading frame changes, and hence the protein changes, and hence the phenotype changes. Now the same thing, okay. Now this was deletion, correct? Now let's say there is an addition. So what I'll do is, I'll get back the C. Okay, so I'll again write down the sequence here, the normal sequence. And let me get C back here, because originally there was a C, correct? So I'm getting C back here. So C back here. Now, now let me do an addition here. Okay, so addition would be what? Let's say if I place G here, this is an addition, correct? One more base has been added. Now, because of this addition, obviously, okay, so there will be a G here also. Now, this is CAU, this is UUU. So now look at the reading frame. It's going to be AUG first, and it's going to be GCA, which is going to be the second codon, then UUU, okay, the third codon, and then it's going to be UUG, this is going to be the third codon. So don't you think the entire reading frame has got again changed because of an addition? So if methionine and let's say GCA, let's say this codes for isoleucine, UUU phenylalanine, okay, and then let's say UUG, let's say this codes for some X, amino acid. So again the protein, the entire reading frame has again got changed. So additions also lead to frame shift mutations, okay. Okay. So when it comes to, one what you have is induced mutations and one is spontaneous mutations. So this is again a different criteria based on when they take place. Okay. So one of the criteria for induced mutations would be, okay, induced mutations. These are caused because of a carcinogen or a mutagen. So mutagen is nothing but any agent. This is an agent which causes mutation. Okay. For example, if you use some chemicals, or because of, let's say, because of viruses. Okay, if these cause mutation, we call them as induced mutation because of a mutagen. And spontaneous mutations would be they occur randomly, automatically, anytime. So spontaneous does not have a reason as such. So spontaneous mutation, they do not, there is no cause as such. They just, occur naturally. They occur naturally. So during transcription, translation, if there is changes in the machinery, then automatically there are changes. It's not because of a mutagen or some, external mutagen or a molecule as such. So mutagens are molecules which induce mutation. They create mutations. Now, second, what we are learning is, okay, chromosomal aberrations. These are chromosomal aberrations. Now, before that, let's do substitution. I think first what we have finished is, we have finished only gene mutations, right? And frame shift we have finished. B, let's start with the B one. Okay. This is substitution. Substitution. Substitution means replacement of one base. This is replacement of one base with another. Okay, with another. Now there are two types. First is under this transition. Okay, transition. So when you have, okay, a purine, when a purine, which is A or G, is substituted by another purine, or a pyrimidine. Pyrimidine is P or C. This is substituted by another pyrimidine. Okay, so you have purines and you have pyrimidines, correct? Now, out of these, you have A, T, G, and C. Now, purines, if you have a purine which is substituted by another purine, or a pyrimidine by another pyrimidine, these are called as transitions. This is transition. So one purine is replaced with the other. So instead of A on the DNA, if there was G, and if there was T and T was replaced by C, or C by T. So purine by purine or pyrimidine by pyrimidine, that is called as transition. And second one, what you have is transversion. Transversion is wherein, okay, you have a purine which is replaced by a pyrimidine. This is transversion. You have a purine which is replaced by a pyrimidine. So these will be transversion. Okay. So purine with pyrimidine or vice versa.